going to now introduce Hal Collard, who I think you're going to talk about how the next session is going to go.
Yeah, that's right. Can you hear me? Yeah. Hey, Kevin, before you leave, I wanted to make a comment to the group about the registry. So hi, everyone. I'm Hal Collard. I'm from California, the University of California in San Francisco.
And he's really tall.
Yeah, that's right. Well, I wanted to rank centers by height.
Yes.
Right. I just wanted to, Kevin may not say this, so this to me is really a transformative development in the field, this registry. So I just wanted to acknowledge that. I mean, I don't know how many of you were at the last summit. Maybe you could raise your hands, those of you who were at the last summit. So it looks like maybe, I don't know, a third of the group or something. So you may remember this was, I think, just being launched. In fact, I was looking at the date, November 15th. Maybe that was the summit. I can't remember, but it was right around then.
Yep.
So this has really been wonderful to see and never could have happened without the people in this room. So I just wanted to thank all of you, the foundation for doing this, and really also you, Kevin, for organizing this and leading it. It's been and continues to be a real contribution. So we're all, speaking as the ILD community, excited. So thanks.
No, thanks, Al.
It's a lot of work. It's a lot of work.
It's a lot of work that's done by a lot of phenomenally really good people. I'm just the one that's lucky enough to come up here and talk about it, but this is a team project from the foundation, and your sites, if they weren't involved in getting the data to us and all that, we wouldn't have any of this. So this is really, it is transformative in a way that we're working together as a community. Yeah. So thank you.
Yeah, and I wrote down the first goal. That's why I'm looking at my phone. I'm not checking my texts or, right. The first goal of the registry was create a cohort of well-characterized patients with PF for participation in research. And I just wanted to emphasize the participation word. It's a two-way participation. I mean, we really are looking to the patient community to engage, not just through providing information, but ideas and energy. And I hope as this goes forward, there's increasing opportunities for direct patient engagement and thinking about what priorities we should be looking at, how we should do that, helping us figure out the best ways to do it. I think that's one of the big goals of this registry. And in clinical research in general, things are really finally recognizing that voice is really important. So this is another great example of a community and engagement that way. So thank you, Kevin. Okay, so it's great to be here. This is my favorite conference, really, because it's unique in its bringing together the patient community and people like us. And I'll just reiterate Kevin's opening comments about how meaningful it is to us to be here. Please stop us and talk to us and ask us questions if we don't do that the same to you. We really are all here to interact, and so please feel free to come up at any point and introduce yourself and ask questions. It's welcome. So this panel discussion is to talk about the basics of research, and we framed it around how a drug gets developed. There are many aspects to research, and thought this was one way we could kind of talk about the different levels of or stages of the research process. We're each going to talk for about 5 minutes. I tend to talk too much, so I probably have already taken up my 5 minutes. But we're going to talk for 5 minutes each, and then we really wanted to have an interactive discussion. We came up with some questions for each other, but would love to hear from you as well. So with that introduction, I want to ask John Morthanos first to come up and speak to us. John is a PFF Ambassador and is going to tell you a little bit about his experience with research. Thanks, John.
Hi, everybody. For those of you who don't know me, I'm John Morthanos. 5 years ago, September, I had a lung transplant at New York Presbyterian Columbia, and things are going well. Oh, and one important thing is, remember www.pulmonaryfibrosis.org. From there, you can get to find out about the Care Center Network, programs that are going on, support groups in your area, all the information you need. In the year 2000, I began finding symptoms taking place, and it wasn't until 2010 that all the other pulmonologists I saw who said I was obese, asthmatic, allergic to cats, and ugly were found to be wrong because I was told at that point I had IPF. And that was at Yale University. Yale is a center, one of the centers of excellence, but they don't do lung transplants there. And they referred me to Columbia New York Presbyterian for the transplant. So I was seeing two sets of doctors, with Columbia being my primary. And Yale was involved in a few drug studies, drug trials going on, and they asked if I wanted to participate. One of the things, when you have a disease, and as many of you know, it's not a very pleasant disease, and what they tell you, without a transplant, it's a very ugly life situation. And they said, you want to participate in this study? The first question is, you don't know if you're going to get a placebo or you're going to get the drug. Am I going to live long enough to see if it's working? Is it going to help? Isn't all that? And you quickly overcome that because as you're sitting in support group meetings, you see new patients coming in, new families coming in, new caretakers coming in, and you look at the fright in their eyes and you go, if I can participate in a study now, and if that drug tends to help or they find that it's helping, it will help other people. I'll be doing something good for the future. So the study I was a part of was an infusion study. It's still going on. I believe it's in its third life cycle, which means that it's been successful so far. But it was abruptly ended for me because I was fortunate enough to get a lung transplant. But hopefully whatever results I was going through, that the 3 hours of infusion that I went through every 3 weeks, that the CAT scans and the PFTs I went through every 4 weeks, all led to the step 2 of that study, which said, these drugs are helping, what else can we do? So as a patient, as a lung transplant survivor, as your friend, I would say if you have the opportunity, look at doing this study. If you can become part of one, you have to ask your doctors what's going on, if it's available. Also, the Pulmonary Fibrosis website will list what's going on, where you can sign up. You're not doing it to get rich, you're doing it to help yourself and to help others. And I guess that's all I have to say, so thank you.
Thank you, John. I'm just going to switch this slides. We have just a few slides to show you here. David, which one? What are they called? This one here. Yeah. Great. Okay. Just trying to advance the slides. There we go. Okay, I have 2 slides for you today. So I'm mostly going to stand here away from the lectern and try to just talk to you about some of the basics of drug development. So, and actually John mentioned this about the different stages or phases. This is, I like this figure. I know it's, I walked back to the back of the room and it's hard to see this from back there. Apologize. It shows the process from left to right of drug development, and it starts with the first column, which says basic research. I would just call that research in general. It's a pretty narrow column, but it's actually a huge enterprise, and that's something we can talk about in the Q&A if you're interested in going into that. That's a registry, for example, is a big part of the type of research that goes into identifying where we start looking for drug candidates. So that is really a vast operation. Next to that is drug discovery. So there is a process once you understand where to focus your energies and research of screening for candidate compounds or drugs. And that is a complicated and expensive process and often leads to many potential candidates that seem to have some affinity for impact on a mechanism that you think is relevant to the disease. Sometimes that is jumped over a little, and things are just tested directly in models of disease and looking for impact. But ideally, you have some kind of mechanistic-based discovery, and that's a huge focus, as David maybe talked to, of pharmaceutical companies and academics, and it's a lot of work and a lot of investment. And as you can see from the figure, if you can't see the details of the words, you can see that wide white range there that narrows down over time. So there are lots of candidates at each stage of this that drop out. So you go from the drug discovery then to the very last or preclinical stage, where you take your potential compound and you look to see what its toxicity is, what its tolerability is, what its kinetics are, like how well is it absorbed, does it reach the levels or reach the tissue you think is important, and then most importantly, is there evidence either in the lab or sometimes in models of disease for an impact and efficacy. And as many of you probably know, this is a challenge in lung fibrosis because we haven't had very effective, or at least we think effective, preclinical models. So we haven't had a model in which we're pretty confident that if we show an impact, it's going to translate into an impact in the human disease. That's not unique to pulmonary fibrosis, but it is a real challenge of ours. And it's hard for drug developers because if you don't have a good model, you take a bigger risk because you can't have as much of a sense that this drug is going to have the impact that you hope it does. But once things make it through those stages, you're now into the clinical trial development. And this is where typically there are 3 stages or levels of trials, 1, 2, and 3. Very quickly, stage 1 is essentially a safety and sometimes dose-finding study. Some of you maybe have been involved in these. Sometimes they're a single dose, sometimes they're multiple doses, but they're pretty short and they require a lot of blood draws and check-ins. Once a compound is established as safe and a dose is found, phase 2 is really a more full-fledged tolerability study and a preliminary efficacy or effect study. So ideally, at the end of phase 2, you have a very good sense for the tolerability and safety of a drug and its likely impact on the disease. And this is where, for example, the drug compound you were involved with is at currently. And once that looks promising, and promising is really in the eye of the as I say, in the eye of the beholder. There's no great definition for what promising is, and this is one of the challenges developers face. What level of evidence do you need to make that final investment into the phase 3 trial, which is where you establish true efficacy and ultimately, if it's a new compound, regulatory approval and the ability to provide it to patients. And as you can see, that narrows down to a very fine line, and after FDA review, essentially, multiple compounds in multiple years and thousands of patients have gone into developing and delivering one drug. So this is what we have. We can talk about actually some ways in which the FDA and others are thinking about trying to make this more efficient. There are good reasons for all of these stages, but it's slow and expensive and long. And clearly, we need to do everything we can to accelerate it. My second slide is a much briefer discussion just to show you the money behind it. And the main point I wanted to make with this slide, again, if you can't read it, there's no way you can read it. The red largest part of this pie, it says 64%, this is expenditures in biomedical research by year. So 2/3 of the expenditures are industry, and about 1/4 are the federal government. That's a lot of that is the NIH, but there are other agencies that invest significantly in research. The Department of Defense does, the Department of Energy does, the FDA does, among others. The federal government makes a big investment in biomedical research. So this is a partnership between government and generally academia and industry, and could not function without all of those components. And really, it works extremely well, in my opinion, across those groups. And there are other important contributors, but the pie sizes are much smaller. So this is just to imprint the actual amount of money spent. That's $120 billion a year in biomedical research, and that it really is split primarily between government sources and industry. Okay, so I'm going to turn it over to Dave, and he's going to talk a little bit more about development, and then we'll have time for conversation and questions.
Great, thanks, Al. My name is David McNinch. I'm kind of talking to you wearing two hats today. One is I'm on the board for, there you go. All right, thanks, Scott. Scott's right here. He's never going to let, he's going to help manage me. One's from the standpoint of the board of directors of PFF. I've been fortunate now to be on the board for 3 years. And the other is I've been in the kind of biopharma industry for a little over 25 years. And prior to coming onto the board, I was with Intermune, and we had a lot of ambitions and things that we were doing and working closely with PFF. So I'm going to, as I answer questions, I might go from one thing, kind of one perspective to the other. Before I pick up where Hal left off, I did want to say one thing about the registry and the care center network. Just real quick, if we look back at just 2013, we were funding the potential idea of a registry and a pilot of 9 care center networks. And here we are 4 years later with 42 care center networks and a registry that's recruiting beyond expectations. And I can assure you that the dreams and ambitions of the clinical team and PFF as a whole are far greater than what you've seen now. So Scott and Bill Schmidt, our new CEO, and our development team are working diligently at finding ways to raise money to increase the size of the registry, the things that we can do. So what I would say is if you're not able to kind of enroll right now, don't get too frustrated, because I can assure you people are working very hard to kind of continue to evolve. And if we can make the same kind of progress we made in the next 4 years as we have over the past 4 years, it's going to be tremendous. So that's the side. But I want to pick up with where Hal, and I wanted to point out something that's interesting. And when you think about orphan diseases, the Drug Orphan Act was passed in 1983, and since then we've had 525 new drugs approved in the U.S. For rare diseases. And that quite honestly didn't pick up until maybe 20, 25 years ago. And prior to that, industry investing a lot in orphan diseases, if you wanted to see drugs developed for a rare disease, you had to raise the money yourself. And obviously one of the great examples of that is cystic fibrosis. Bob Bell, was a pioneer in raising money to invest in cystic fibrosis drugs. Fortunately, and it comes with some drawbacks, but fortunately now industry is investing in rare diseases so much more significantly, so that it's not on patient foundations to do that. So if we looked at that, if we looked at this pie chart and kind of focused it on rare disease 30 years ago, industry's investment would be very narrow compared to what it is today. So one of the things that is changing is, when the cost of drug development. And there are a lot of good reasons for it, but when you include the failures, and there are far more failures than there are successes in drug development, but when you include the failures of drug development, today, it takes a little over $2.5 billion to get a drug to market, and when you consider the failures. And there's a lot of reasons for that. One is we're, in the past 10 years, we've started investing a lot more in innovative medicines. And the more innovative medicines are, quite honestly, the more failures you're gonna have, but we don't need the 15th statin to come to market. Thank God we're getting away from a lot of the Me Too drug development. And we're getting a lot more, it's more complicated. We're collecting more data points. We need more patients. Some of the trials are getting bigger. So there's a lot of reasons that drive that cost, but it's a very expensive endeavor when you look at drug development. And it's also, it's also a community. It takes I was going to put the line on top, is it takes a village. It's not just a drug company or just the investigator. When you look at all the people that are involved and all the, from the nonprofits to the clinical coordinators and the CROs, the pharmacists, the NIH, academic institutions, clinical trial sites, FDA, it takes all of these people and all of these institutions working collaboratively to ensure that we have success. And that's just to get the drug developed, much less to get it utilized and managed and paid for appropriately. Finally, to put some real kind of numbers to the, also the expense, if you look at, in general, for what percentage of drugs that get into phase 1 actually make it to approval, it's 8.4%. And if you take away the ones that had biomarkers, it's more like 6%. So when you think about that, it really is a high-risk business, and the odds are low. But one of the things that if you look at this, what the blue bar shows, that if you have a biomarker, a validated biomarker, you go from 6% really to over 25%. So you got 26% of the drugs that have validated biomarkers then make it to approval. That's right there. If you look at, so what's tangible that's going to eventually come out of this registry? One of the things that we'll be able to understand from the blood draws and different things that we're doing and different things that we hope to do is the more we understand about those patients, the more we can understand validated biomarkers or other approaches that will put targeted people and targeted therapies into those clinical trials, which will yield faster clinical trials and higher likelihood of results. So, so those are the points that I wanted to leave you with. The other thing that I would say is, if you looked at this slide and said, hey, let's just look at how many different programs and how many different companies have investments in IPF. If you looked at that in 2013, the logos would be a lot bigger because there'd be a lot fewer companies. And I'm sure I've probably missed one or two companies, but if you just look at the companies as well as National Heart and Lung and Blood, as well as NIH, there are a lot of people now investing in IPF and other PF-related diseases, and that is fantastic, because the more you have, as we go back to the less than 1 out of 10, the more shots on goal you have, the more opportunity you have to have success. So I think, I wish Rome. I remember when I was living in Boston, they had the big dig was going on, and there was a big, right when you got into the construction, it was miserable. They said, Rome wasn't built in a day. If it had been, we'd have hired their contractor. So drug development doesn't happen overnight. But when you look at the kind of progress we've made in the past 4 years, you start getting very optimistic over the next 4 years and 10 years. And it's the work that we do today that will make the differences tomorrow. So that's the end of my piece. And so I think we're open to what questions you have.
Great, make me live again. Thanks, Dave.
Sure.
So, so we're just, let's take questions. And again, we have some general topics, but I'd love to, hear from you. So I think at the front table here, go ahead.
Oh, my sister right now is at the ER for something. She did do her wrist, but she's the one that's got pulmonary fibrosis. She's had it for 3.5 years. And for 2016 and 2017, she's been on Fibrogen. And now they're going to stop her from it, kicking her off of the third phase. Why is that?
Yeah. Well, I can take a stab at that. I don't know as I know the specifics, but let me talk just generally to the issue of investigational drugs in clinical trials and what happens to patients afterwards. I think there's always a balance. So the drug is of unknown efficacy, that we have to say first. So we don't know if the therapy is helping or not, but that's why we're doing the trial. But we have some suggestion that it may. And I'm speaking generally here, not about the Fibrogen product per se. That's just the nature of these trials. In some instances, sponsors decide to continue patients on what's called an open label extension, which allows additional safety and tolerability information to be collected, and if the drug is beneficial, allows patients to stay on it. There is the other alternative, though, that it could be that the drug is either not helpful or potentially harmful, and so continuing patients on therapy after a trial before you know the answer could actually be exposing them to more harm. So there are discussions around this often in the ethical review, human subjects review committees at institutions about what's the right approach. I think there are pros and cons to both. From a purely scientific perspective, it's undeniable that for a patient, engaged in a trial, we all have hope that these compounds work. I think probably 9 times out of 10, at least in my experience, patients would like to stay on the therapy and know that they're getting active therapy. Not the right word maybe, but a benefit of being in the study, to have something that may be helping them. I don't know what the right answer is to that overall, but I think that maybe I've at least illustrated the reasons why sometimes studies will stop it and sometimes studies won't. And just looking, you have another question, then I do want to ask Dave if he has any comments.
So if she has to, sorry, she does have to stop it now, and what space of time does she need to take before she can go into another trial? I mean, are they going to let her know, or?
Yeah, usually it's not particularly long in between. She should be eligible for another trial, either
like
that
in a month?
compounder. Usually it's 28 days or so.
Okay.
It depends, but it's usually not very long.
Okay, great. Thank you.
Yeah, maybe in the back, a man in the gray
Oh.
sweater there. Oh, sorry. I was. Yeah, you have the microphone. Go ahead. Yeah.
Oh. All right. Well, I'm just wondering about the European model for bringing a drug to market. Is it substantially different than what happens with us? It's very similar then.
Very similar. It's very similar. The European authorities will have their own views on endpoints and what's appropriate. More often than not, they're aligning with FDA. One of the things, Europe has actually been a little more proactive, especially in rare disease areas, of having early approval that will be they call it a contingent approval. So it's based on maybe phase 2 data and with the ability, if the validating study doesn't prove out, they can pull it back. So Europe is being a little more proactive on that, but then same challenges. Europe, the authorities don't want to pay for those drugs that they consider experimental.
My second part of this is, because you keep bringing up rare diseases, is PF really that rare?
No. Well, that's a great point. So because it's an orphan, so orphan drugs were defined as patients with a less than 200,000 in the U.S. And so so rare disease, orphan disease, it is, but we're all aware that it is not as rare as some people think it is. So it's we believe.
I just think that might be a good way to change the conversation.
Fair enough.
It's not so rare.
Yes, other questions? Okay, how about in the gray sweater there, sir? I'll repeat your question if you can't get the microphone.
Thank you very much. My name is Michael Harrington. I lost my grandfather, my father, and my uncle to IPF, and my brother and I have been tested for the TERT mutation, and so that's the reason why I'm here for my first summit today. It's also my birthday, so I was going to see if I could have 3 questions, but they'll be very specific, so I think they'll go by quickly. Hal, you mentioned in the preclinical phase, you said that one of the challenges is that you need to have a good model, right? And I was just wondering, could you just comment a little bit more on what helps to create a better model in those earlier phases? So that's question 1. Question 2 is, on your slide, you had the nice kind of multi-phase process. For the clinical trials through FDA approval, could you give some sort of sense of time? I mean, best to worst in terms of years, realizing that there's not, a perfect answer there. And the third question goes to, I think it was David's slide, when he talked about a validated biomarker seems to improve the success rate. And I was wondering if you could just, or maybe this is a dumb question, I just didn't know what a validated biomarker was. So just if you could provide additional commentary on that.
Okay, I can.
You hit all 3.
I can comment on the first 2 quickly. Usually we talk about preclinical models. Do I need to speak louder? Yeah, okay. Yeah, usually we talk about preclinical models. So a lot of these are animal models. In a lot of diseases, that's where the real signal as to likely efficacy comes from, and there's at least reasonable correlation between success in an animal model and success in a human disease. So really that's been the quest, is to find something that in an animal model would predict efficacy. And I would say, in general, we've been unsuccessful with it. What to do about that is a difficult question. I mean, developing a new model, it's a lot of people working on it, but I don't think there's any easy answer to that. I can tell you one thing that the field has recognized, though, is that studying as much as possible the human disease, meaning cells or tissue from people who have the condition, and understanding mechanism, at least the mechanisms that are relevant to disease through the study of the actual disease tissue, might be a more effective and efficient way to go, and actually might point us in the direction then of how to develop models more that are more useful. So that's a work in progress. And the timeline for development is usually, I mean, most of these slides say 5 to 7 years. I think it depends on the nature of the question and the disease, particularly in diseases that are less common. There can be slow recruitment, longer trial duration needed depending on what measurements you're looking at. So it all plays into it, but the average, you're talking about up to a decade sometimes for that process. I mean, hopefully shorter, but so it's not quick. Dave?
So yeah, so related to validated biomarkers, thinking about the best way to answer this, probably one of the best way, very easy thing to understand is who in here is on a statin? Anybody raise your hand? Sorry. You don't take a statin because you want lower cholesterol. You want lower cholesterol because you want to prevent a heart attack, right? So if you think about the work that was done to understand the role of cholesterol, drug trials were a lot faster and a lot smaller to show that you can lower the cholesterol, but you only did that because you had to understand broader what was the role of cholesterol in heart disease. So that's, to me, that's it goes back 30 years ago, but that's a really tangible example of when you find a biomarker that's validated, how you can then have faster and more impactful drug development.
Great. Other questions? Yeah. How about in the blue top? Right. Oh, or not? Okay. Yes. Great.
I have a question, but it's not about drugs that can help pulmonary fibrosis. I was diagnosed with pulmonary fibrosis, but it was ruled that it was caused from Vicks Vapor Rub.
Yep.
And I was wondering, I've looked into trying to find other people that's been affected like me to start the process of maybe getting action against the company, so the other people won't develop it like I did. So I just wanted to know, when a drug causes the disease, what would you need to do?
Yeah, I'm glad you asked that question. It's a really difficult area of lung fibrosis, is figuring out when a drug is responsible for the disease. It clearly happens. There are clearly certain drugs that we know are more commonly associated with it. I think the best, but then there are a lot of situations where it seems to be temporally related, but it's just not something that's well described. There is a great resource that is directed at physicians, but I think is actually very useful for patients as well, online, called pneumotox. I think it's pneumotox.com. It's organized by a French group, and it goes through the literature and puts together the reported complications, lung complications of various drugs, and it gives some sense of the nature and frequency and kind of strength of the association. I find that very useful for me because this comes up a lot where there's associations that we've not reported, and it's always hard to know if they're true and unrelated or if they're causative, and that can help, I think, give some information. So that might be a place to start, and then always reporting associations through your physician is a good way to get them recognized.
My pulmonologist said he'd seen it in textbooks, but I was the first case he'd seen in person. He had heard of it, and he said it was Vicks Vapor Rub and fish oil. So, I mean, it's called lipoid pneumonia, but it caused fibrosis in my lungs.
Yes, right. That is described, right, from oils, essentially,
from the
inhaling
petroleum
oils.
jelly and the
Right.
and the Vicks
Yep.
and other things, camphor oil and turpentine oil and stuff. So thank you for answering my question.
Pneumotox, this is your channel, pneumotox.com.
Okay.
And there's an app too, I think, yeah.
Yes, at the summit in La Jolla 4 years ago, everybody was so excited because the two first drugs were about to be approved by the FDA that could maybe help some patients, Ofev and Esbriet. And since then, there's not been a lot of hoopla, waiting for something else to be approved. I'm just curious, are there any drugs in clinical trials now that you specifically, any of you up there, are watching curiously with anticipation of anything? And you can't say all of them. Is there something that's caught your attention?
Well, I guess, the way I'd probably characterize that is, what are the things that are making it past phase 2 and into phase 3? The one, I think there are 2 or 3, but I know, for instance, the fibrogen drug should start phase 3 trial. So I think, the way next year, and then I would expect that trial to probably take 2 to, if we use Esbriet as example, 2.5 years or something to that effect in total. What else is out there that is going to phase 3?
Yeah, no, I think that's, I would answer the same way. And in general, when I'm asked about that, this is a problem in the field, is that kind of ranking or triaging the likely benefit of therapies is really difficult because we don't have a good model in which to base that. What I look for is a mechanism that's known and that makes sense. So we call it a biological rationale. So a good biological rationale, and ideally some proof that the drug actually affects that mechanism in people and that it's safe and tolerable. I think if you have those things, and I guess the other thing is the trial is looking at something that's important to patients, then to me, that's about as much as we can ask for, unless you then get a drug to the stage, but I think fibrogen is a good example of that, where you now have some real evidence from trials that there may be a benefit. Yeah, how about right here, sir? Yeah.
Are you doing much on genetic engineering as opposed to drug?
I'm not necessarily the right person to ask, but I don't think so. No, we're not really. I don't know of any genetic engineering. There are some pharmacogenetic approaches that are of interest. In other words, looking at people's genetics and tailoring therapy to their genetic profile. And there is a study, for example, that some people here are developing, that are at this conference are developing, looking at actually at N-acetylcysteine, which was tested in IPF generically, but looking at it in a subgroup with a certain genetic, with a genotype that looks like it may have a benefit. So there is work going on in the kind of pharmacogenomics, but I don't know of anybody who's doing any actual engineering or approach to that.
Yeah, the only thing I'd add is, we're looking at potential things for the registry to further understand genetics of patients that are currently suffering from IPF. So the more we can understand that and start segmenting that, then you can really start have a much better understanding of which type of patients and what their genetics role is, which is kind of the first step to then, okay, is there a way to intervene? So I think we're years away from that second part, but if we can start more, we can understand about the genetics of IPF patients and what we can learn from that, then that puts us, the first step down the road.
Yeah, and genetics, just say, can be, to just expand on that, can be a really important guide as to where to focus. And one of the notes I made to myself that I wanted to try and mention at some point was I really think we should think as upstream as possible when we think about therapeutics. At least so in other words, as close to the root of the problem as possible. There can be a problem up here that then causes this and this and this, and your therapy may deal with this kind of end stage or downstream issue, but particularly in a complex medical condition like fibrosis, where there are lots of ways that can go and it's hard to block it the further down you get. This is conceptual, but I think targeting things further upstream could have a big bang for the buck, and genetics help point us towards where the potential root problems can be. So I'm hopeful that that'll be, as we understand more about that, and we really are understanding a lot of the genetics, that'll drive therapies that can target those areas. Yeah, I don't know who would be, yes, great, the microphone, yeah.
Can you talk about combination therapies such as OFEV and Esbriet and other things that are being considered or in process?
Yeah, I can speak to that. Yeah, combination therapy, probably the one that comes up the most is the combination of the two approved therapies. So we don't really have any efficacy data yet on combination therapy, but there has been a study published recently that looked at safety and tolerability of combination therapy. This trial was done in Europe. And I always remember, I think it was patients who were on stable nintedanib and had they were stable, I think, on nintedanib or OFEV and had the second drug, Esbriet or pirfenidone, added. I may have it backwards, but I think that's right. And essentially, my takeaway from that was that it was safe. And while there was some increase in side effects, it was not intolerable for many patients. So it seemed to be certainly a reasonable thing and manageable for a lot of the patients who were engaged in the trial. It was a shorter trial than typical efficacy trials, and it was smaller. They did look at lung function, and there was less decline in lung function in people on combined therapy than on monotherapy. But the numbers are small enough that there is definitely a risk for error in that estimation. So I've taken away from that, and I've had a few conversations with patients about this, that in my personal opinion, we don't know if the combination works yet, but there's at least some data that suggests it might. It seems safe, and its tolerability on average is reasonable, and of course you can stop the combination if you don't tolerate it. The big barrier there to me is cost, at least for a lot of patients, and access. But I think it's very reasonable to consider it if you and your clinician feel like the monotherapy is not sufficient. But recognize we don't know yet really about whether it's effective, truly effective.
I've got the mic
Yeah.
back here. What are the present-day possibilities of developing a drug that actually reverses the scarring in pulmonary fibrosis?
Oh, how's it going to be? It's funny, for the longest time, I think most people thought that was completely impossible. But if you talk to some of the researchers now, they have reasons to believe that it may be possible. Whether we've actually seen a drug that can, do that, I think is aspirational. And I'm not aware of any research programs in that, but but I've talked to Dr. Cosgrove about this, and and he's a believer that that may be more possible than people think, but we're just nowhere near it. I don't know how's a much better scientist.
Well, I think that in certain diseases that may be true that we can reverse some of the fibrosis, but part of the problem, particularly in more advanced fibrotic diseases, is not just the scar, but the destruction of the underlying, we call it the architecture of the lung. So the normal anatomy is no longer there. So in that case, even if you were able to remove the scar, there would be no functional, you're not going back to a functional lung there. So I think this was another point I wrote down to make, is that what does cure look like? I think that for me, cure is really returning of lung function. That's what we want. That's our goal, is to get people's lung function back to normal. The therapies we're looking at currently, at least for a lot of the more fibrotic conditions, may only get us halfway there. They may be able to stop progression, but not necessarily return function. And it could be that we need a completely different approach to the return in function need. And that's where regenerative medicine comes in potentially. Or, let's think creatively. I mean, maybe there are, technological ways in which we can improve gas exchange and oxygenation that doesn't require more tissue regeneration, just requires better efficiencies. But those two questions in my mind are often, I think, may require different approaches, I guess is the point, and we have not focused as much on that second part, which I think for many patients is a central need, particularly if you have more advanced disease. It'd be great to stop it, but you still have a lot of impairment, and we need to fix that. Yeah, great.
Hi, Dr. Collard. I have a couple questions. One is kind of a dumb one, but I don't know the answer. On the slide you had on the current expenditures on biomedical research, you said the 64.70% was industry. What is industry?
Oh.
So when you think of industries, basically small companies, large companies, biopharma companies.
Okay, so those are the pharma. The second question I had was on the slide for the phases of clinical trials. Now, I'm a big proponent of clinical trials as long as I understood it. I don't understand the medical jargon. And by the way, I'm Cindy from Texas. I suffer from scleroderma, and then as a result, 35% of my type of patient also have the ILD. So mine is interstitial, not idiopathic. I know exactly where it comes from. So I'm huge on clinical trials. I really am, because I already know what my outcome is, okay? So what I like to know in specifics is how long, and you said 5 to 10 years on the entire phase. Well, how long is phase 1? How long is phase 2? How long is phase 3? And as it narrows down, are you losing people from death or really truly quitting? Because I knew through some of the trials, I could quit 6 weeks in, and I kept going even if I had symptoms. Secondly, my big thing was I didn't want to have the placebo, okay? But in a blind study, you don't get that choice, okay? So going through a study or a trial, if it had worked on me like it did on several other patients, why can they not stay on the drug as it goes through the FDA process and it's helping them? Why can't they stay on the drug? And last, when it is not approved by the FDA, does this start back all over again from phase 1? And, you know, with IPF or even my own illness, you can go at any time. They gave me 12 years, but I'm on year 17.
Yeah.
So do you see what I'm saying just from a patient standpoint? Those are issues that we are concerned about.
Yeah, I think there are a lot of points you made there, which are all excellent, and I don't think I can address all of them, but I'll just say that, part of what you came back to was that when you feel like there's a benefit, why can't we keep people on these therapies, either waiting approval or after the trial? And I think, as I mentioned before, there's just if a therapy was clearly and measurably beneficial to patients, there are compassionate use programs and things that often will get people access to that. Unfortunately, we've dealt with a lot of uncertainties where we've had to wait for the study results to come out because the impact on individuals is much less clear. And so, there I think what we talked about earlier, the balance is just different people or groups decide different things, and we don't know the right answer.
So maybe I'll just add two quick points. One is, because it goes back to the discussion about open label studies. Each company has to make that decision on open label. It's assuming that we know that it believes it's safe. And open label trials or keeping people on post phase 2 is a very expensive endeavor. And some companies choose to do that and are able to do that. Some companies don't, and which is very difficult for patients, I think, and I'm certainly one that would vote normally to continue that if at all possible, financially feasible, because patients like John, like yourself, have invested your time and effort into the clinical trial and certainly deserve a result. So the second point is, I think you brought up is, as drugs, how do you get additional indications? So usually what will happen, it's rare that a lot of times drugs won't be in development for many different indications before they come to market. And usually they'll go, okay, we have one indication, now the investment is a lot easier and the hurdles to getting approval and doing trials in other diseases are a lot narrower because you don't have to go back and find out if it's safe, right? So let's say if there's a drug for IPF that we thought might work in scleroderma, then you know the drug is safe, so you only have to do an efficacy trial. So that makes it a lot quicker.
So I think in the interest of time, I think I want to make a comment, and then I have a question actually for John and David maybe to summarize. And my comment is that those of you who have other questions, please remember what I said at the beginning. Please come up, and whether it's me or someone else who's here on the faculty, and stop us and ask us your questions, and that's what this is all about. I'm sorry we're short on time here, but I do want to maybe close with a question to you that may summarize Kevin's and our conversation, which is if you could give advice to the foundation or the people in this room about how we can make a difference or where we should focus our energies to move research forward, what would it be? Would it be engaging in registries that get you potential access or what?
So
what would you say?
Just the one thing I would say is together as a group, we can do tremendously more things than we can as an individual. And never think of yourself as being alone. And I think that's what PFF as an organization, whether it's patient education, whether it's patient support groups, or whether it's clinical research, be as engaged as much as possible with PFF, because there's no better organization that is looking across that gamut and working with industry, working with FDA, working with pharmaceutical companies. So stay as engaged as you can. And for the record, Scott's not here, is engaged as also if write a check so for PFF. We appreciate that because the more we can do. So that's probably the quickest, best answer I can give. And again, if you feel like you're alone, find ways to connect with other patients, and then you can find out, because there's nothing, it goes back to misery loves company. Whatever you're miserable about, the weather, whatever, there's someone just like you that's dealing with the same issues, and the more you spend time with that person, the better you'll feel.
And John?
Again, support groups are key to working with others, getting answers for yourself. Not to give up hope, but unfortunately for those of you that don't live near a center of excellence, or your center does not do studies, find out where and if there's a center that you can travel to, because many of the studies don't require you going in every 3 weeks for a 3-hour infusion. It could be taking pills at home, it could be taking a liquid, and you may be able to participate as long as your doctor is able to do the testing requirements to feed back the researchers if there's improvement or any sign of cessation of the continuation of scarring.
Great.
But it's really to ask questions, and don't let area limit you if you can.
Great, thanks John. Well, David and John, thanks for joining me today, and thank you all for being here, and we'll look forward to seeing you around the conference.